Hello students, let us start with chapter 6, tissues. What are tissues? Tissues are a group of cells with similar structure and they work together to achieve a particular function. Now what are the differences between plant and animal tissues? Plant tissues are dead cells and they are supportive in function, and the maintenance of these cells is less. The reason is plants are stationary or they do not move, while in animals there is constant movement. Therefore, cells are living. There is wear and tear. Therefore, maintenance is required in the case of animals. Next, difference in growth pattern. In plants, the growth usually occurs at stem and root tips. But in animals, the growth is uniform throughout the body. Another aspect is plants grow throughout their lifetime, but animals grow only up to a certain age. Next, regarding the structural organization of organs and organ system is highly specialized in case of complex animals. But in complex plants, the specialization is quite less. Let us now discuss the different types of tissues that are found in plants. Plant tissues are broadly classified into two: permanent tissues and meristematic tissues. Now, meristematic tissues are again subdivided into three, that is, apical meristem, intercalary meristem, and lateral meristem. Permanent tissues are again divided into two, that is, simple permanent tissue and complex permanent tissues. The simple permanent tissues are further subdivided into parenchyma, collenchyma, and sclerenchyma, while the complex permanent tissues are made of two types of tissues, that is, the xylem and the phloem. Let us now discuss about meristematic tissues. Meristematic tissues have cells that are constantly dividing. They are very active cells. They are having dense cytoplasm. The nucleus is prominent and they are relatively unspecialized cells. Not only that, they generally do not have vacuoles. Now children, can you tell me what might be the reason? In chapter 5, we have discussed the role of vacuoles. One of the functions of vacuole is storage. Now here, in meristematic tissues, as I told you, they are constantly dividing cells. So there is no need for any kind of storage. Since storage is not required, vacuoles are generally absent in meristematic tissues. Let us focus on the different types of meristematic tissues. They are apical meristem, intercalary meristem and lateral meristem. Apical meristem is usually found in root and shoot tips. Therefore, its function is elongation of the root and stem. Intercalary meristem is found at the base of leaves and in internodes. Therefore, it helps in the elongation of leaf stalk and also elongation of the stem. The lateral meristem is also known as the cambium. It helps in the increase in the girth of the stem and it is found in the circumference of the stem. Let us refer to activity 6.1. We need to take 2 onion bulbs and 2 glass jars and fill it with water. Label them as 1 and 2 and place the onion bulbs above each of the jar. We keep this experimental setup as such and observe the growth of the length of the roots every day. When it is day 3, we take each of the bulb, measure the length and make a note of the length of the roots. We will find that the length is almost the same in both the jars 1 and 2. After measuring, we have to cut the root tips of the onion bulb that is kept in jar 2. Then again, like in the previous, keep the experimental setup for another 2 days. On day 5, we take the bulbs and once again measure the length of the roots. We will see that the length of the roots of the onion kept in jar 1 has increased further, but there is no increase in the length of the roots of the onion kept in jar 2. Can you find the reason for this? Children, I have already told you where do meristematic tissues, where are they present? They are present in the root tips. And you know that they are constantly dividing cells. So if these tips are cut off, that means we are removing the meristematic tissues from the roots. Therefore, the growth is stopped. It does not increase. Therefore, we can conclude that meristematic tissues found in root and shoot tips help in the elongation of roots and stems. Let us now discuss what are permanent tissues. Permanent tissues are formed from meristematic tissues by a process known as differentiation. These tissues, they lose their capacity to divide and achieve a particular shape, size and specific function. This process of achieving a particular shape, size and function is known as differentiation. Thus, permanent tissues are formed. Now, permanent tissues, as said earlier, are of 2 types. Simple permanent tissues and complex permanent tissues. Now, what is the difference between simple permanent tissue and complex permanent tissue? Simple permanent tissues are made of a single type of cell, while complex permanent tissues are made of different components. Let us discuss the different types of simple permanent tissues. First of all, parenchyma. They form a few layers of cells and it is the basic packing material of the plant. They are relatively unspecialized cells. The cell wall is made of cellulose and it is thin. They are living cells and there is lot of intercellular space. They are found in stems and roots. Their main function is to store food and water or nutrients and water. They also help to provide support to the plant. When these parenchyma cells contain chlorophyll, they perform photosynthesis. Such specialized parenchyma cells which contain chlorophyll and can perform photosynthesis are known as chlorenchyma. In certain aquatic plants, along with the parenchyma, in between they have large air spaces. This helps to provide buoyancy to the plant and thus the plant can float. This type of parenchyma cells are known as aerenchyma. Next, let us discuss collenchyma. Collenchyma provides flexibility to the plant. It helps in the bending of stems and leaf stalks without breaking. It also provides mechanical support to the plant. They are living cells elongated with little intercellular space and they have irregularly thickened corners in the cells. Next is the sclerenchyma. Sclerenchyma provides the plant stiffness and hardness. It is found in the husk of coconut. Sclerenchyma cells are dead cells. There is no intercellular space. The cell wall is highly thickened due to the deposition of a chemical known as lignin. They are found around vascular bundles in stems, veins of leaves and also the hard outer covering of nuts and seeds. They also provide mechanical support to the plant. With that, we have come to the end of today's session. Thank you.